Greetings, biochemistry students. This is a lecture for Chapter 14 of Lehninger's Principles of Biochemistry. This is Dr. Michael Garut at Missouri Southern State University. You will note that this is not the exact same PowerPoint that comes with the 8th edition. I've taken out some things, added some things that I think are important, and some things from previous editions. This chapter primarily focuses on glycolysis, which is how we get energy from glucose. It also talks about fermentation, gluconeogenesis, and a little bit about the pentose phosphate pathway. Glucose is a good fuel because it's quite soluble, it's easily transportable, and you can save it as glycogen, so it's quite easily storable, and because you can use it to make most of the other biomolecules that you need, such as lipids, amino acids, and nucleotides. The four fates of glucose are: if you have plenty of energy, to store it as glycogen. If you don't have plenty of energy, to oxidize it to pyruvate. And the other ones are synthesis. So we can use it to synthesize the things needed by the cell, or we can synthesize 5-carbon sugars. This pathway also produces NADPH for what we call reducing power, and we'll talk about that later on. In glycolysis, which is normally considered the 10 steps from glucose to pyruvate. Pyruvate is a useful biological intermediate. At the end of glycolysis, however, you've only produced a couple of ATPs and a couple of NADHs, so you have a little bit of the free energy captured in these energy-carrying molecules, ATP and NADH. Glycolysis is expressed in pretty much all organisms and all cells of all organisms, so it was one of the earliest pathways studied in biochemistry, and discovering that ATP was sort of the central energy carrying molecule. We usually think of glycolysis as starting from glucose specifically, so here's your structure of glucose. If you don't start from glucose, then you can either convert the molecule to glucose, so a different sugar, trehalose, or sucrose, which can be converted into glucose plus fructose, and then fructose can enter glycolysis after step 2. You can also get to glucose from its storage in starch, that would be here. Other sugars either convert to glucose or enter glycolysis at different points. This is the same diagram from a different edition of the textbook, so if you're looking through the PowerPoint, you can pick which one you like better. I actually like the old one better. When you cleave from glycogen, you get glucose 1-phosphate. Here are some examples of some disaccharides that break down to glucose or other sugars like fructose and galactose, and those, as we saw in the previous slide, enter at different points. Those monosaccharides can be absorbed directly in the small intestine. As we've talked about in a previous semester, animals do not have the enzyme necessary to break down the beta 1-4 glycosidic bonds that are present in cellulose. And so if you eat paper, we just get fiber out of that. We don't get energy out of that. Even though it's made from glucose, we cannot separate those from the polymers. I don't find this something that's necessary to memorize, but it's just interesting to see that we know how sugars that are not glucose are converted into things that can go into glycolysis. In the case of fructose 1-phosphate, that breaks down, as you know, in step 5 of glycolysis into 2 3-carbon sugars. One of those is glyceraldehyde 3-phosphate. Well, if you end up with glyceraldehyde that's not glyceraldehyde 3-phosphate, then you need an extra enzyme, triose kinase, to attach an ATP onto that so you can get the glyceraldehyde 3-phosphates, which can enter glycolysis. There are some pretty common disaccharides that are in dietary molecules. Milk sugar is called lactose, and there's an enzyme that all infants have that converts lactose to glucose and galactose, the monosaccharides. Often this enzyme is diminished in amount or completely absent in adults, which gives the lactose intolerance. This is an overview of how glucose gets into the cell and is used for energy. So if you start outside the cell, there's this membrane transporter, for example, GLUT4 transporter, and glucose gets transported into the cell. Once it's in the cell, it gets phosphorylated by the enzyme hexokinase or maybe glucokinase to make glucose 6-phosphate. Now that it is phosphorylated, it's no longer a substrate for this transport protein, and so it doesn't get back out of the cell. Then it can go through glycolysis to make pyruvate. Pyruvate ends up being oxidized via the citric acid cycle, and most of the ATP come afterwards in oxidative phosphorylation. So like 30 ATP roughly from that, whereas you only get 2 from glycolysis. The other parts of this diagram are about the regulation of the glucose uptake and the insulin response, which is not the focus of this chapter. In general, the strategy of glycolysis is to activate it by phosphorylating it. This gives it the so-called, I'm doing air quotes now, high energy phosphate bonds, and then to begin the process of oxidizing it and breaking it into smaller molecules, conserving some energy as ATP and NADH. Here's a picture of the 10 steps of glycolysis. You probably found one, maybe this exact one for your pre-activity, or maybe some different ones. You can't really see this, it's too small. So here it is broken down into the 2 phases. So we can call the first 5 steps the energy investment phase or the preparatory phase. In this phase, you start with a 6-carbon sugar, you phosphorylate it, you isomerize it, and you phosphorylate it again. So then you have fructose 1,6-bisphosphate, which it now has 2 high-energy phosphate bonds. This is useful because now when you split it into 2 3-carbon sugars, each of those has that high-energy phosphate bond. And these 2 sugars are pretty interconvertible by the enzyme triose phosphate isomerase, which is considered to be one of the so-called perfect enzymes in that its rate acceleration is so fast that every molecule of dihydroxyacetone phosphate can go ahead and make glyceraldehyde 3-phosphate. Then you have the second half, the energy producing phase or the payoff phase. In this case, there are, again, 2 steps that produce ATP. However, since you now have 2 glyceraldehyde 3-phosphates, 2 3-carbon sugars, then you get to count each of those twice. So you're actually making 4 ATP, leading to the net gain of 2 ATPs. There is only one step in glycolysis that is an oxidation. It's step 6. You can tell it's an oxidation in a couple of ways. One is that it uses the cofactor NADH, and another is that it has the enzyme has the name dehydrogenase. This reaction does add another phosphate, so you might think it was a transferase, but since it is a redox reaction, this is an oxidoreductase, and the energy of that oxidation is actually saved in this high-energy phosphate bond. In the next step, you take that high-energy phosphate and you transfer it to ADP to make ATP, which is the energy currency of the cell. Then we do another isomerization. Then we have our second high-energy phosphate that we use to make an ATP. We have to do an elimination first. So we eliminate an OH and an H from these 2 carbons, which gives you an alkene, but the alkene has an oxygen on it, and if I'm going to cleave off this phosphate, then it's going to be an enol or an enolate, and you'll remember from organic chemistry that for ketones, the keto form is much more stable than the enol form, and so that simply tautomerizes, and that also adds to why this is a very exergonic reaction, because the enol is not stable, whereas the keto form is stable. Now you have 2 pyruvates and 2 more ATP. So again, we've taken a 6-carbon sugar to make 2 3-carbon sugars. We have made a net of 2 ATPs by phosphoryl transfer, and we have made one molecule of NADH for each of the two glyceraldehyde 3-phosphates. This is a diagram from your textbook that describes basically what I have just described. So if you want a nice summary of that and the rationale for the steps, that is there for you to look at. Now we go through each of the steps individually and hit some highlights. The first reaction is called hexokinase. Hexo because it works on hexoses. In other words, it does not just phosphorylate glucose, but also can phosphorylate other 6-carbon sugars. There are actually different hexokinases. This says irreversible. In class, I would, instead of saying irreversible, I think we generally call these far from equilibrium steps, which makes them essentially irreversible. When you have this very negative, like more than negative 15-ish or so kilojoules per mole, you're getting into the far from equilibrium territory. In eukaryotes, the enzyme is hexokinase. In other organisms, there's a different enzyme that's called glucokinase. And then it turns out that different tissues have different isozymes of hexokinase. Humans encode 4 different hexokinases. That means they are isozymes. They're expressed in different tissues. And they have slightly different specificities, but that is not the focus of this chapter. The next step is an isomerization. The enzyme is called phosphohexose isomerase or phosphoglucose isomerase, or sometimes glucose 6-phosphate isomerase. You can see why. And this shows you the mechanism of that. So there's a 4-step mechanism if you don't include just the E binding to make that enzyme substrate complex, right? So enzyme substrate complex not shown here. But you take this and you make the hemiacetal into the open chain form. And what we need to do to isomerize glucose to fructose is to move this carbonyl group from carbon 1 to carbon number 2. That's all that needs to be done. So that happens by a general base catalyzed step where you pull the hydrogen off of carbon 2 and you make an enol. And then a general acid catalyzed step where you donate the hydrogen back to carbon 1 and it gets back to the keto form. So that's a tautomerization. And then you recyclize it back up into the fructose 6-phosphate. In step 3 then, you start with the fructose 6-phosphate and you use another ATP to do another phosphorylation of carbon number 1 to get fructose 1,6-bisphosphate, which is, again, a far from equilibrium step. Because it's far from equilibrium, that's essentially irreversible. It's also considered to be the first committed step to glycolysis. In other words, once you've gotten here, since that's basically irreversible, it's committed to go through all the way to pyruvate. Now you might be thinking, why is step 1 not the committed step? And for that, we need to look at some other pathways. Step 1, which is glucose to glucose 6-phosphate, is not the committed step because glucose 6-phosphate has other pathways. For example, this pentose phosphate pathway or storage as glycogen. Whereas once you get to fructose 1,6-bisphosphate, there are no other side pathways for this to be utilized, and so it's the committed step, committed to glycolysis. That enzyme, therefore, is a key enzyme for regulation, because since that's the committed step, if you downregulate that, then you're going to downregulate the entire glycolytic pathway. So this makes sense. If the ATP is depleted, you would want to activate that enzyme, and if you have excess ADP or AMP, that would also activate it. This would be the energy charge of the cell is decreased. When that's the case, then you're going to want to activate PFK1. There's also this other allosteric activator, fructose 2,6-bisphosphate, which we'll talk about when we get to the regulation chapter. And it can also be indirectly activated by ribulose 5-phosphate, which is an intermediate in the pentose phosphate pathway. Now you have a reversible step that cleaves the fructose bisphosphate into 2,3-carbon sugars. This is the reverse of an aldol condensation that you learned about in organic chemistry. There are two different kinds of aldolases. The ones in animals and plants use covalent catalysis. The ones in fungi and bacteria use metal ion catalysis. And I thought I deleted this little comment because I don't really like pull reaction forward. So we actually talked about this in class. About 95% of the pool of these two sugars is in the dihydroxyacetone phosphate form because it's more stable. Ketones are more stable than aldehydes, if you remember that from general chemistry. However, since glyceraldehyde 3-phosphate is the one that's used in the next step, then this reaction pool just reestablishes equilibrium very quickly, and even though only 5% of it is in this form, that does not limit the flux or the rate through the glycolytic pathway. Okay, so returning to the mechanism, it says something here about the Schiff base intermediate. A Schiff base is what in organic chemistry we called an imine. An imine is a carbon double bonded to a nitrogen, and a Schiff base specifically has to have an R group attached to the nitrogen. That makes it an imine that is a Schiff base. So once the fructose bisphosphate has been ring opened, you have this covalent attack of a lysine on the carbonyl, facilitated by general acid catalysis of some group on that enzyme. And then we're headed towards this imine or Schiff base formation. First, you have a general base that removes a hydrogen from that lysine, causing that double bond to be formed between the carbon and the nitrogen. So that's your Schiff base, but it's in the protonated form. So then in that protonated form, it's particularly unstable, and you can then have the bond cleavage between these two carbons facilitated by a general base, removing this hydrogen, making the double bond, the carbonyl, and then kicking out half of that substrate as a leaving group, which is now an ene-amine instead of an imine. You got the alkene singly bonded to the nitrogen, so that's an ene-amine. Then this next step, which it says is an isomerization, I think really is better defined as an acid-base reaction, because what you have is this general acid donating a hydrogen to this carbon of the alkene part at the same time as you're reforming the double bond from the nitrogen to make the imine. So I don't think that's an isomerization, because it could be a tautomerization if you transfer a hydrogen from one place to another on the same molecule, but this is actually an acid-base reaction. Again, you get to this protonated Schiff base, which is not particularly stable. In other words, it's high energy or activated, and that allows water to come in as a nucleophile to attack this carbon and displace the active site lysine. There are a lot of mechanistic steps there that are not shown. That would be the hydrolysis of an imine, which tends to come up in mechanisms at the very end of organic chemistry too. The following slides here show the same mechanism, but just blown up into larger. So if you would like to replay the audio that I described while looking at the PowerPoint and scrolling through these so that you can see them larger, that would be fine. Then we get to step 5 of glycolysis, the conversion between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Those are the two components of what had been the fructose bisphosphate. It's now split into 2,3-carbon sugars. As we talked about in class, the molecule that proceeds through glycolysis is the glyceraldehyde 3-phosphate, but this equilibrium highly favors the dihydroxyacetone phosphate. That is because ketones are more stable than aldehydes, as we learned in organic chemistry. So it says this is reversible, which it is, but 7.5 kilojoules per mole difference means that the the product and reactant distribution is like 95% to 5%. That's not a problem because you can, once you use up some of this 5% to go on into the next step, this reaction to isomerize them is extremely fast. In fact, this enzyme, triose phosphate isomerase, is thought to be one of the so-called perfect enzymes. It's perfect because its rate is only limited by how quickly the substrate, the DHAP, can diffuse into and out of the active site. This is called diffusion controlled, and it is considered to be the maximum rate that an enzyme can possibly go because every molecule of reactant that comes into the active site converts to products. You then have this pool of metabolites, but only the glyceraldehyde 3-phosphate continues through. These now, once you get here, are completely indistinguishable from each other. So if you had any kind of isotopic labeling, it's going to be scrambled between these two because of that equilibrium. This slide shows what I was just already talking about. And that completes the preparatory or energy investment phase of glycolysis. Now we proceed to the payoff phase. Step 6 is the only redox, the only oxidation reaction in glycolysis, and it takes glyceraldehyde 3-phosphate, attaches an inorganic phosphate. So you might think, oh, well, that is a phosphorylation reaction, so that would be a kinase. But no, because what you're actually doing is you're using the energy from this NAD going to NADH reduction to do an oxidation of glyceraldehyde 3-phosphate. You can tell it's an oxidation because it was an aldehyde, and now it is an ester, a phosphate ester. But the shortcut for organic compounds is the more bonds you have to oxygen from a carbon, the more oxidized that carbon is. So this carbon has 2 bonds to an oxygen, and this carbon has 3 bonds to an oxygen, so it's more oxidized. So at this point you have conserved much of the energy of glucose in NADH, which has a high reduction potential, and also in this acyl phosphate, a quote high energy phosphate bond. Those acyl phosphates, as you can see there, have a very high standard free energy of hydrolysis, which is the formal way of saying high energy phosphate. And we know it's high because the hydrolysis of ATP to ADP plus a phosphate has a standard free energy of hydrolysis of about negative 30.5 kilojoules per mole. So that means this is significantly higher than that, meaning that once you put a phosphate there, it has enough. When you couple that with the conversion of ADP to ATP, that's still going to be favorable. This says it's an indirectly energy yielding because it's giving you this NADH, but you can't recognize the energy output of NADH until that proceeds through the electron transport pathway. We will investigate this reaction in class specifically, and you'll notice that the diagram in our workbook looks a whole lot like this diagram, except that in our workbook I think it shows it as having 6 steps. And now I see why the author of that workbook put the NAD and the NADH where she did, because that's exactly where this author put it. But notice that the actual conversion of NAD to NADH happens here in step 3. And so what the textbook author is, I assume, trying to show here, and it's indicated by in this yellow box, is that the NADH leaves the active site, and the NAD comes back into the active site. We don't, as biochemists, typically use arrows like this to indicate substrates dissociating or associating. That's just not how we tend to use these curved arrows. These are almost always intended to mean reactants or products of that step, and that's not what happened here, so that led to my confusion. So this reaction does produce 1,3-bisphosphoglycerate, which has this high energy acyl phosphate, which then in step 7, that high energy acyl phosphate can be used to, as I mentioned, phosphorylate ADP to make ATP. If you take the negative 49.3 and you subtract 30.5 from that, you get about 18.5. Well, it's actually 18.8, but that's pretty close. This is called a substrate level phosphorylation, and we're going to see why it's called that in the next slide. So it's a substrate level phosphorylation because it's a phosphoryl transfer from a substrate, as opposed to a phosphate just being added directly from an inorganic phosphate. That would be an respiration-linked or oxidative phosphorylation. So a substrate level phosphorylation is what this is when you take the phosphate from a higher energy phosphate bond to a lower energy one. And again, when I say high energy and low energy, what I mean is the very high standard free energy of hydrolysis, right? Okay. So now, if you recall, the triose phosphate isomerization, that is a positive 7.5 kilojoules per mole. That's why we only have roughly 5% of this pool in the glyceraldehyde 3-phosphate form. But when you take that positive 7.5 and add it or couple it with this negative 18.5, okay, within error 7.5, 18.5, that gives me a negative 11. They're saying a negative 12.2, so there must be a little bit of rounding off error there. But the gist of it is, if you consider these two reactions together, it's overall favorable. So even though step 6 is unfavorable, step 7 is so much more favorable that makes the equilibrium from step 6, the glyceraldehyde 3-phosphate, all the way through to the 3-phosphoglycerate. It looks like the 3 was lost here. Makes that favorable. Then we have step 8. The 3-phosphoglycerate isomerizes to 2-phosphoglycerate, so you can imagine that is pretty energy neutral. 4.4 kilojoules per mole is near equilibrium. Your book shows a little bit about how the mechanism of this works. You have a histidine acting as a base, pulling off a hydrogen, and then you have that oxygen on carbon 2 acting as a nucleophile to hit the phosphorus and kick out another histidine residue. So we've got a general base reaction and a nucleophilic reaction. Does this happen simultaneously? It doesn't say, but that is what it shows. It seems odd that it would, but presumably they have not drawn this in a realistic orientation for that to happen. And then you just reverse those steps so that you can then put the phosphate group back onto carbon 2 and you have completed the isomerization. Step 9 is called enolase. Again, this is a near equilibrium step. It's a little bit unfavorable because an enol, right, here we have carboxylate and then an alcohol. This is called enolase because you get to something that has an alkene attached to an oxygen. If this were an OH, that would be an enol, hence enolase. But this is now like an acyl phosphate. It's an enol phosphate, and that is also an activated or high energy phosphate. So that can then be used to transfer that phosphate onto ADP to make ATP and still be very favorable in terms of the standard free energy change. This is the same slide with a different image, right? So here's one image and here is a different image, but it's the same slide. When you hydrolyze off this phosphate, you end up with enol pyruvate, but the enol form is much less stable, and so that tautomerizes to make the keto form that you see as pyruvate. Here is a summary of the overall reaction of glycolysis. You took glucose, which had 6 carbons. You ended up with 2 pyruvates, which have 3 carbons each, and you have a net generation of 2 ATP and 2 NADH. So overall, even if you divide the glycolysis into the 2 phases, the preparatory phase and the payoff phase, and one phase is very exergonic and one is somewhat endergonic. Overall, the free energy change is negative 85 kilojoules per mole under standard conditions, and even under cellular conditions, glycolysis is very favorable, essentially irreversible, meaning that once you've produced pyruvate, NADH, and ATP, the equilibrium is very much going to favor those products as compared to the glucose that you started with. Once you get to pyruvate, you can use that pyruvate to be further oxidized for energy, which happens via the citric acid cycle and the electron transport oxidative phosphorylation. You can also use the pyruvate for you can use it for synthetic purposes, or you can use it for anaerobic fermentation. Here are the three things that you can do once you get to pyruvate. So under aerobic conditions, the next step is pyruvate gets converted to acetyl-CoA by the pyruvate dehydrogenase complex, and at that point the first carbon of glucose that leaves as carbon dioxide. So actually the first and the second carbon, one from each of the two pyruvates. That makes acetyl-CoA, which is a reactant for the citric acid cycle, and after the citric acid cycle, the rest of the carbons of the glucose are released as carbon dioxide. The next thing you can do with pyruvate is you can use the pyruvate to make either lactate in some organisms or ethanol in other organisms. Both of these are called fermentation, and the rationale for why this is necessary is if you don't have oxygen present, then these pathways become blocked for reasons that we'll see later. And when they become blocked, what happens is pyruvate can't go to acetyl-CoA, but more importantly, all this NADH that is being produced cannot be converted back into NAD in the mitochondria by reacting with oxygen. So you have this buildup of NADH and you use up all of the NAD resources. If that is unchecked, then glycolysis is going to stop because it requires NAD for step 6. So under anaerobic conditions, in order to keep glycolysis going, you have to regenerate that NAD in some way. And the way that is regenerated in most organisms like animals is that pyruvate is converted into lactate and the NADH is oxidized back to NAD with the reduction of pyruvate to lactate. And that's quite favorable. So that then takes the NADH you made from glycolysis, recycles it back into NAD so that glycolysis can continue. But the only way that works is if you just continue to build up lactate indefinitely. At some point, it becomes very difficult to continue to build up lactate because, yes, this is very negative, but once you get larger concentrations of lactate, that lowers the actual delta G of that reaction. So that is called lactic acid or lactate fermentation. Fermentation is a general term that means some process that helps to extract energy without consuming oxygen. So if you consider the entire reaction of glucose through pyruvate to lactate as a process that generates energy, that would be considered fermentation, lactic acid fermentation. Yeast can also ferment instead of making lactate to make ethanol and carbon dioxide, which is the feature of yeast that allows them to make alcoholic and carbonated beverages at the same time. So I mentioned that lactate cannot be made indefinitely because what happens is within a minute or so the lactate builds up so much that makes it more acidic, and it also shifts the equilibrium of that pyruvate to lactate, what we call a dead end pathway. It's a dead end pathway because you can go in to lactate and the only way out of lactate is to go back to pyruvate. that has to happen during a recovery phase. So you need some time to recuperate from this lactic acid fermentation, and that happens in the liver in something called the Cori cycle. This picture is from a different chapter of your book, but you have lactate buildup in the muscles that goes out into the blood and to the liver, and then you can take that lactate, reverse that back to pyruvate, and do gluconeogenesis to get to glucose, which can then go back out into the blood and back to the muscles to supply them, and they can then store some extra glucose as glycogen. This is called the Cori cycle, and this takes time. I asked Google, and so I only have a limited amount of faith in this, but it says that takes about an hour to diminish the lactate concentrations. It's a common misconception that it's the lactate buildup itself that causes the muscle soreness, the lactate buildup and the lowering of the pH. But what we actually think is the cause of the muscle soreness is the fact that the muscles have run out of energy to some extent and therefore become damaged. So if you have soreness after a workout, it's not because of lactic acid production, it's because of actual cell damage that has taken place, physical damage or damage from low amounts of ATP, which also damages cells, specifically cell membranes. So here are the reactions of ethanol fermentation, pyruvate being decarboxylated to make carbon dioxide and acetaldehyde, and then that acetaldehyde reacting with NADH and being the acetaldehyde being reduced to ethanol. It's interesting why humans have this enzyme, alcohol dehydrogenase, when we don't have this enzyme, pyruvate decarboxylase. We can easily break down acetaldehyde with an enzyme called aldehyde dehydrogenase to make acetic acid or acetate, which looks very much like acetyl-CoA, which we can metabolize to carbon dioxide and water, no problem. So it seems that the only reason that we have this enzyme is that we must have evolved to a need to be able to metabolize ethanol, presumably in food that had been fermented that otherwise would not have been able to provide energy or be metabolized, like if the ethanol was not able to be detoxified by this pathway, then those foods would be a lot more toxic to us than they are. The enzyme pyruvate decarboxylase is one that we will look at specifically later. It uses a cofactor called thiamine pyrophosphate, which you see here, and that cofactor increases the nucleophilicity of the enzyme. That's not important for glycolysis. It's not important for this chapter particularly. This is the structure of thiamine pyrophosphate. Thiamine is vitamin B1. Thiamine has this thiazolium ring. Thio means sulfur and azo means nitrogen, so thiazolium. That ring has a hydrogen on it that it is in between, on the carbon, in between the nitrogen and the sulfur. That is an interesting position for it to be in, because it makes that hydrogen much more acidic than you would normally expect a hydrogen on a carbon to be. I believe the pKa of that is around 19, which is pretty similar to the acidity of water or alcohols, and not at all similar to the pKa of 40 or 50 for alkanes or alkenes. That means you can have a reactive carbanion at that stage, and if you look at the reaction pathway, that's what you see. You see this hydrogen removed by some group, doesn't show what, but that's your active carbanion, and then that becomes a nucleophile that can react with the pyruvate. You then have a decarboxylation, you have protonation, and then you have an elimination, so pyruvate ends up becoming acetaldehyde. This cofactor from thiamine is important in several enzymes, not just pyruvate decarboxylase, which humans do not have, but also pyruvate dehydrogenase, which we do have, which is the enzyme that converts pyruvate to acetyl-CoA, so that's extremely important. And also one of the enzymes in the citric acid cycle, alpha-ketoglutarate dehydrogenase, they react by very similar mechanisms with that thiamine pyrophosphate. And then there's transketolase, which is an enzyme in the pentose phosphate pathway. So gluconeogenesis, which literally means new birth of glucose, right? Neo is new, genesis is birth, and glucose. Creating glucose from lactate or pyruvate or amino acids or glycerol, but typically we think of it as starting from pyruvate because we typically think of glycolysis as going from glucose to pyruvate and gluconeogenesis as pyruvate to glucose. But there are other routes into phosphoenolpyruvate. We're specifically going to look at right now the exact opposite of glycolysis being gluconeogenesis. But it's not the exact opposite because 7 of the steps are near equilibrium, but 3 of the steps are not. We've looked at this in class. So the 3 steps of glycolysis that are far from equilibrium is step 1 and step 3, both of which are far from equilibrium because they are hydrolyzing ATP. And then finally step 10, which is phosphoenolpyruvate to pyruvate. That one is very favorable because of the high energy nature of phosphoenolpyruvate. Those steps are essentially irreversible in the those steps are essentially irreversible in glycolysis. So in order to make gluconeogenesis happen, there need to be other pathways that are not irreversible. It takes 2 enzymes to get from pyruvate back to phosphoenolpyruvate, and it takes 2 equivalents of ATP. 2 ATP for 2 pyruvates, and there's 2 GTP. So for 1 pyruvate, it would be 1 ATP and 1 GTP, which are essentially equivalent in energy. You can see why this would be. If it's very favorable for 1 phosphoenolpyruvate to transfer a phosphate to ADP to make an ATP and pyruvate, well, it's going to be very unfavorable for that to go backwards. But if you can hydrolyze 2 high energy phosphates, that's how you can make that favorable. You do that by, in the middle, having this oxaloacetate. So you actually take a carbon dioxide or a bicarbonate ion, which is a source of carbon dioxide, and you attach that to pyruvate. So it has 3 carbons. You attach that, it now has 4 carbons. But now you've made a high energy carboxyl group, and that decarboxylates, and that energy is saved in the phosphoenolpyruvate. So it takes those 2 steps. Steps 3 and 1 are bypassed simply by not trying to remake ATP, but just hydrolyze the phosphate away, and that makes those reactions exergonic. Now what you don't want to do is cycle around those reactions. So you don't want to take fructose 6-phosphate plus ATP to make fructose 1,6-bisphosphate and then turn right around and hydrolyze off that phosphate to make fructose 6-phosphate again. That just has a net effect of hydrolyzing ATP for no reason. That is called a futile cycle, F-U-T-I-L-E, futile. And so we're going to see how you avoid that. The main way of avoiding it is by regulating these enzymes separately so that they will be active not at the same time. Here are in red, the far from equilibrium steps. These are the standard free energies of hydrolysis. These are the free energies of hydrolysis under typical cellular conditions. So those are the steps that must be bypassed. Once you bypass them, then you can just add up the total reaction of gluconeogenesis, and you'll see that it's not exactly the reverse of glycolysis. You'll recall that glycolysis has a net production of 2 ATP and 2 NADH. If you look at gluconeogenesis, it's actually requiring 4 ATP and 2 GTP, so that's 6 ATP equivalents. And it's also requiring, not producing, but requiring 2 NADH. That makes gluconeogenesis energetically expensive, because it's if you made pyruvate from glucose, you would net 2 NADH and 2 ATP. If you reverse that, you're going to use up 4 ATP extra, because you produce 2 and you use up 6, and then the NADH, you would have lost a net of 4 NADH because you don't get the 2 in step 6, and it takes 2 more to reverse that. So it's energetically expensive. In order to bypass step 10 of glycolysis, you need 2 different enzymes for that, and you're producing this oxaloacetate and then the phosphoenol pyruvate. So that means there's 1 different intermediate on the way back. 11 steps instead of 10. Pyruvate carboxylase is actually in the mitochondria, and PEPCK, phosphoenol pyruvate carboxykinase, is typically in the cytosol. So that presents a little bit of a problem because those 2 steps are in 2 different locations. Glycolysis happens in the cytosol. Gluconeogenesis happens in the cytosol, with the exception of this pyruvate carboxylase step, which is a mitochondrial enzyme. We'll talk about that part in a second. Pyruvate carboxylase requires this cofactor biotin, which is like a vitamin. And that cofactor biotin, you can see what that looks like here. It has a long tether that literally takes the carbon dioxide that is now attached to it as a carboxylate group and swings it around into a second active site, so it can take that carbon dioxide and attach it to a pyruvate through an enolate intermediate. Imagine you have pyruvate and you remove the hydrogen from the terminal carbon and remove an H+, that then makes an enolate, which is analogous to an alpha carbanion, and that then can nucleophilically attack the carbon dioxide that in this site 2 to make oxaloacetate. You've now added this carboxyl group onto pyruvate, and you have a beta keto acid, right? This is your acid, here's your keto, and alpha, beta. Beta keto acids undergo decarboxylation quite readily. So you've basically produced a high energy intermediate. Instead of a phosphate group, it's a carboxyl group, but that's what you've done. In the next step, you take that intermediate and it does this decarboxylation, and you use that to attach a phosphate from GTP onto the oxaloacetate, which then loses the carbon dioxide and becomes phosphoenol pyruvate. So you take this high energy decarboxylation plus this high energy hydrolysis of GTP, and together those you can create this phosphoenol pyruvate, which is now a glycolytic intermediate and can proceed back up through glycolysis in a reverse direction to make glucose. This picture briefly shows, and it leaves out a lot, but it briefly shows the sort of pathway that has to happen. If you've produced pyruvate in the cytosol, that pyruvate has to get transported into the mitochondria in order to do the first step there, the pyruvate carboxylate step. However, there's not an oxaloacetate transporter. So now you have to have another reaction. You have to have a malate dehydrogenase that converts oxaloacetate to malate, and then the malate can be transported, and then you have a malate dehydrogenase that reverses that reaction in the cytosol to get the oxaloacetate outside. So you have not really transported oxaloacetate. You've converted it to malate, you've transported it, and then you've converted it back to oxaloacetate. That may seem circuitous, and in fact it is, There are reasons for that which are evolutionary and also have to do with regulation of pathways that we'll see later when we look at citric acid cycle and some other pathways. As we mentioned, steps 1 and 3 of glycolysis are also bypassed in gluconeogenesis. You have very favorable steps because all you're doing is taking a high energy phosphate like fructose bisphosphate, chopping off a phosphate to get back to fructose 6-phosphate, and that is far from equilibrium and favorable. And then glucose 6-phosphate to glucose hydrolyzing is also favorable. So as we mentioned, gluconeogenesis costs a lot of energy, but it is essential because you need glucose. So if your glycogen stores run out, then the brain, and especially the red blood cells, the erythrocytes, they don't have mitochondria, so they can only get energy from glucose. So when your glycogen and glucose are depleted, then somehow we need to make glucose. So even though it costs some energy, it needs to be done. We have seen that we can make glucose in gluconeogenesis from pyruvate, but anything you can make into pyruvate also works, like lactate or oxaloacetate. And it turns out, as we'll see later in the class, that you can turn many amino acids also into oxaloacetate or pyruvate or things that can make oxaloacetate or pyruvate. So we can make glucose from carbohydrates or from proteins. We cannot make glucose from fatty acids because once we get acetyl-CoA, we do not have the right enzymes to convert acetyl-CoA to any intermediates that add carbons to it that can get to phosphoenolpyruvate and produce glucose. We'll see why that is later when we talk about the citric acid cycle. Finally, the pentose phosphate pathway is one of those 4 fates of glucose, and we're only going to talk about that very briefly here, but here's an overview. So once you get to glucose 6-phosphate, the first step of glycolysis, it has other fates. One of those is to, instead of proceeding through glycolysis, getting to step 6 and making NADH, we have a slightly different step which produces 6-phosphogluconate and NADPH instead of NADH. NADPH is exactly like NADH except for a phosphate group, but that makes them recognized differently by enzymes. So interestingly, the ratio of NAD to NADH is maintained pretty high. Think about the reason that we need NAD is so that step 6 of glycolysis can continue. The reason we need lactic acid or alcoholic fermentation is to regenerate that NAD anaerobically so that glycolysis can continue. Conversely, the ratio of NADP to NADPH is very low. We keep the ratio so that we have a higher concentration of NADPH than NADP, and that is because we use NADPH for anabolic reactions, these reductive biosynthesis reactions where we make things like fatty acids or sterols or anything else we need to make. So we have NADPH used for anabolic reactions and NAD used for catabolic reactions. So that's one of the purposes of the pentose phosphate pathway, is to produce this NADPH for what we call reducing power, the energy necessary for our anabolic reactions. The other product is this ribose 5-phosphate, which you would need whenever you want to make nucleotides or coenzymes like NAD or any of those, FAD. But you would imagine that you need a lot of this for messenger RNA or when cells are dividing to do transcription of DNA. So that would be when this pathway would be more active. Those are the 2 products of the pentose phosphate pathway. And if you take, let's see, 6 times 5 is 30. So if you take 6 5-carbon sugars, that's 30 carbons. You recycle those through a bunch of steps, and you can end up with 5 6-carbon sugars. So what you're doing in these steps, there's a bunch of steps that take 5 carbon sugars and shuffle the carbons around until you have a 6-carbon sugar, glucose 6-phosphate again. Not quite as many of them, but that allows you to then either restart that pathway or do glycolysis or whatever else you need to do with your glucose 6-phosphate. So in summary, we can get a little bit of energy out of glycolysis anaerobically. We can make glucose out of pyruvate, but also out of other things that can be made into pyruvate or phosphoenolpyruvate. We know that steps 1, 3, and 10 of glycolysis are bypassed in gluconeogenesis, and we should know why. And we finally saw that the pentose phosphate pathway produces ribose to make nucleotides and NADPH for reducing power. That's the end of the chapter. It was a doozy. Cheers.